All right, welcome to the third biotechnology screencast. This one covers learning targets 7 and 8, and you're going to follow along in your interactive notebook on page 228, 229, and 230, and get down the information from the slides onto your notes. All right, this first one goes through a timeline of key years in biotechnology history. And the ones that are on the note outline are starred on the slide here. So I'll just go through those ones. In 1866, Mendel published his work on the heredity of peas. 1953, again, Mendel worked, the father of genetics, way before Watson and Crick discovered the structure of DNA. That, remember, again, was done in 1953. 1970, recombinant DNA technology was developed, so using plasmids, restriction enzymes, being able to combine DNA from two different sources. In 1984, DNA fingerprinting developed, so we were able to use gel electrophoresis to separate DNA by size. In 1997, Dolly the sheep was first cloned. And in 2003, the Human Genome Project was completed. And we'll be watching a movie later on about the Human Genome Project, which kind of goes through the history and how that all developed. All right, so this first part is on genetic engineering in plants. The history is kind of, this isn't on the slide, but the history is selective breeding. I mean, for years and years and years, hundreds and hundreds of years, thousands of years, farmers have picked and selected their best animals, their best crops, their best producing plants, and use those to breed for the next generation. Today, what we can do is plants can have favorable traits directly added to their genome. So today, plants can have favorable traits directly added to their genome. So the genetic engineering process produces organisms with new traits. Transgenic is another word for organisms that have one or more genes from another organism inserted in its own genome. So make sure you know, oops, I need my pen again. Make sure you know that word transgenic. One or more genes from another organism inserted into its genome. So this is what I just said about the selective breeding that is still done today. However, today now plants can have favorable characteristics directly added to their genome. So how this is done is it uses plasmids, okay? What we were talking about before, the gene of interest is inserted into the plasmid is inserted into the bacteria, then the bacteria is going to infect the plant, and the new gene is going to become part of the plant's DNA. Just like all the other ones, so that one ends up being expressed just like everything in the normal plant genome. All right, so how have we changed plants? We have made them resistant to frost, disease, drought, and insects. The ability to make natural pesticides. We've improved the nutritional level of plants, and we can more control ripening, so there's less waste of food that becomes too ripe before it hits the grocery store shelves, let's say. And then on the bottom here it says draw a bacterial cell with a transgenic plasmid below. So for a plant cell, well, just draw a bacterial cell. So bacteria cell, it has that extra chromosome, it has the plasmid, and then make sure in the plasmid you draw another inserted gene of interest, just like we did in those other notes. All right, moving on to genetic engineering in animals. Transgenic animals are harder to produce and achieve than plants or bacteria. There's much more to the development, especially mammals. But basically the process, again, you take your gene of interest, you insert it into a fertilized egg. The egg is implanted back into the female. A small percentage of these eggs mature normally, and only a portion of these eggs will actually contain the foreign gene in its DNA. But then all the offspring from the genetic engineered animal will inherit the transgenic trait. So transgenic mice are often used to study human development and disease. So the answer in that one is what type of animal is often used? Mice. The genome is similar to humans, it's readily available, easy to handle, relatively low cost of use, and a high reproductive rate. And there is a think about and discuss question there that I want you to jot down an answer to. Obviously, we really don't need glowing mice. Yeah, they're cool, but the goal is not to make glowing mice so we can make glowing humans. Again, cool, but not the purpose. What would be a useful example of how we as humans might use that technology? So think about that, jot something down, and then we'll move on to genetic engineering in medicine. So genetic engineering in medicine, the goal is to genetically engineer critical proteins that have failed in the body. Genetic engineering using bacteria enables mass quantities to be produced for medicine development, such as anticoagulants, those help dissolve blood clots, insulin for diabetes, and something called factor VIII to promote blood clotting. These next notes move into section 9.5, which is Genomics and Bioinformatics. The Human Genome Project is first. So the whole goal of this project was to map and sequence all of the DNA base pairs in human chromosomes, identify all the genes within the sequences, and store information in databases so it can be used by others. And we've heard about the Human Genome Project in several of the video clips I've shown throughout the genetics project, and I think it's awesome that we have it mapped. It was a big deal at the time, and I think a lot of people thought that, once we map it, we're going to have all these answers and cures for all these diseases. That hasn't happened because we do know that that linear sequence of DNA is only part of the puzzle. It's how those things are then made into proteins and how those proteins interact that really, really is going to give us more information in terms of getting answers to some of these diseases. The project began in 1990. Originally, it was planned to last about 15 years. Some rapid technological advances accelerated the completion date to 2003. And mainly that was in just the DNA sequencing. It was able, instead of being able to do it by hand, it was automated so that machines could do it, so it really sped up the process. Ultimately, because of the Human Genome Project, we have learned that the human genome contains about 20,500 genes, way less than what was originally anticipated. We are 99.9% identical. There's only about 1%, 0.1%, not even 1%, 0.1% of our DNA that is different from one person to another. I think that's pretty amazing when you think about it. We discovered more than 1,800 disease-causing genes, developed more than 2,000 genetic tests for human conditions, and at least 350 biotechnology-based products are currently in clinical trials. And the cost of sequencing an individual's genome has been cut from $8.9 million, I don't think I would have done that if it was $8.9 million, to about $1,000. And there's different companies. I referenced a few of these when we started genetics, 23andMe, and I think there's a Heredity type name of a company where you can have your genome sequenced. And then moving on to section 9.6, this is genetic screening. So this is the process of testing DNA to determine a person's risk of having or passing on a genetic disorder. It uses pedigree analysis and DNA testing. It's used to look for specific genes or proteins that indicate a disorder. And there's currently tests for about 900 disorders, including the gene linked to breast cancer, BRCA1 and BRCA2. We're going to read, I think that's one of the bioethics cases that I pulled that we're going to be looking at Friday and next Monday. So as a preview to those ethics cases that I just talked about, why don't you jot down some things that, in answer to the question, why might genetic screening raise ethical concerns about privacy? This also goes back to kind of some of the things that we discussed when we watched the movie Gattaca. One of these genetic screening tests is for muscular dystrophy. And if you look at this particular section of DNA, if there are missing bands, that indicates that there's a potential for muscular dystrophy. So again, why would, if an employer knew, if an insurance company knew, why might there be some ethical concerns regarding that information? And what are those concerns? All right, last topic is gene therapy. This is the process of replacing of a defective or missing gene or the addition of a new gene into a person's genome using a vector to insert a healthy copy of faulty genes. So again, those vectors could be plasmids, it could be a virus. Many times when you're dealing with human gene therapy, you're talking about a virus instead of a bacterial plasmid used as a vector. So here's kind of a diagram, again, showing what I just talked about, that virus. You disable the virus so it's not virulent or it doesn't cause disease anymore. You put the gene that you're talking about it into that virus, and then the virus is going to go infect the normal cells, and the idea is to get it to correct the stem cells, and then those stem cells, when they regenerate to make new cells for the person, they would have the corrected gene in it. The challenge with gene therapy is the correct gene needs to be added to the correct cells, and we're talking about, hundreds of thousands of cells that need to be corrected. It's not just one. The gene's expression must be regulated so it doesn't make too much or too little of the protein that we're trying to fix, and scientists must determine how the new gene will affect other genes, if that would be an issue or not. Currently, diseases being treated with gene therapy include some cancers, cystic fibrosis, hemophilia, rheumatoid arthritis, and forms of genetic blindness. We are going to read an article dealing with blindness and eyesight in class tomorrow. All right, that does it for the biotechnology learning targets 7 and 8 screencast. Be ready for your 6-point checkup quiz tomorrow. Bye, guys.